My name is Milan Fiala. I am a physician and a researcher at UCLA, and we are developing therapy of sporadic Alzheimer's disease. We are going through innate immunity. I will discuss in the next hour the immunopathology of Alzheimer's disease and the main role of G lymphatic system and macrophages in clearing the brain of soluble and fibrillary amyloid beta. In the future, we have potentially good news for Alzheimer's disease because we begin to understand the defects in the immune system, and we show that some defects are repaired through omega-3 fatty acids. It is still not clear why some patients have better results than others. We test omega-3 in an emulsion called SmartFish, in which omega-3 are protected against oxidation. Our study in patients is voluntary. It's using the SmartFish drink from Norway, and it is approved by IRB. SmartFish drink is supplied by the SmartFish company at a reasonable cost directly to patients who request it. In this slide, we are showing that the immunopathology of Alzheimer's disease involves accumulation of soluble and fibrillary amyloid beta in plaques and around conchophilic vessels and aggregation of phospho-tau. Monocytes migrate across blood-brain barrier into plaques and phagocytize amyloid beta but only incompletely, so that the plaques are not resolved. The approach by Big Pharma has been to enhance clearance of amyloid beta by monoclonal antibodies, but this has largely failed. We take another approach by supplementation with omega-3 fatty acid drink to enhance phagocytosis by macrophages. That means enhancing the innate immunity, not adaptive immunity. Mitochondrial energetic defects in Alzheimer's patients. These defects have been investigated by prominent investigators, including Alan Roses, Butterfield, Zontag, Swerdlow, to name a few. The novelty of our study is the use of omega-3 to repair these mysterious energetic defects in sporadic cases. As I will show you, we found that omega-3 fatty acids repair these energetic defects, in particularly in monocytes and macrophages, and provide energy for phagocytosis of amyloid beta. In this slide, we show that macrophages immigrate into the brain and phagocytize amyloid beta, although this is denied by some investigators, probably because they failed to do immunochemistry of the human brain and rely on animal models. Monocytes may enter into neuropil through lymphatics and directly across the blood-brain barrier in brain microvessels. As we show here in slides A and B, in the upper panel, the monocytes, macrophages in brown enter into plaques which are shown with red amyloid beta. The microglia are distinct from the macrophages. In the lower panel under A and B, we show macrophages which penetrate across blood-brain barrier at microvessels and disrupt tight junction protein Z01. We see this in cross-section in A and in longitudinal section in B. Here we show macrophages invading plaques in AD brain, uploading beta, and migrating to conchophilic vessels. In the upper panel, C, we show large macrophages inside the plaque, and we see they became very large. We call them fat macrophages, and they clear some amyloid beta in a Swiss cheese manner. There is no evidence of dense microglial accumulation around the plaque. In the lower panel under F, we see macrophages in red becoming yellow with amyloid beta in gongophilic vessel, which is green. Here we show macrophages and microglia differences. Macrophages in AD brain have importance for phagocytosis and energy. The monocyte macrophages penetrate the blood-brain barrier and clear and phagocytize amyloid beta plaque, but arrest at blood-brain barrier and apoptose. Omega-3 increase transcription of glycolytic and TCA cycle enzymes. Omega-3 increase energy as ATP-linked OCR. Omega-3 increase phagocytosis. Omega-3 are signaling increasing glycosylation. On the right side, the difference with microglia in animal models shows that microglia show increased autophagy, but energy is also important, and it's increased by cyclokeratin. On the next slide, we show the difference between macrophages from normal controls in A and B and patients in C and D. In A and B, the macrophages from controls download up, phagocytize amyloid beta and fill the whole cytoplasm in B. In A, the amyloid beta eventually gets downloaded into late endosomes, which fuse with lysosomes. In C and D, macrophages from patients do not import amyloid beta inside the cytoplasm. Amyloid beta stays on the surface. Here we show trafficking of amyloid beta in C and D, trafficking in control macrophages, whereas in H and I, amyloid beta stays on the surface, does not import into the cytoplasm of these macrophages from patients. We incubated AD brain sections with monocytes from controls or patients. In the left column, we incubated the brain sections with normal monocytes, which look green under A, but under B and C they become golden because they upload amyloid beta, which is red, and they intrude into the neurons and pull amyloid beta from neurons, as can be seen in D, in Z sections of confocal microscopy. On the contrary, when we incubate monocytes from patients, the monocytes in E still look green, but in F they become shriveled, aggregated, and in G they go into apoptosis. So clearly they do not clear the brain. That's it. We constructed a model of blood-brain barrier with brain microvascular endothelial cells on porous membrane and allowed monocytes to migrate into lower chamber through attraction by chemokines. On the left, we show what we call a fat monocyte with ingested amyloid beta stuck to the top endothelial cells. It cannot migrate. On the right, under C, we can see that the endothelial cells became tall. The monocytes without amyloid beta are migrating through the pores, and the endothelial cells show no disruption. Below it is a transmission electron micrograph which shows the endothelial cells. It shows the monocyte, and in G and H it shows the tight junctions. Next slide. So how do we interpret that omega-3 improve phagocytosis of amyloid beta in vivo and in vitro? We have found that omega-3 increase energy, circadian clock, and mitochondrial cytochrome oxidase. And these are not the only results, but these are the results that play a huge role in the effect of omega-3. We compared macrophages of omega-3 supplemented compared to non-supplemented patients, although this was not a randomized study, and we found that glycolytic and citric acid cycle enzymes were upregulated by omega-3. We found that OXPHOS genes were upregulated by omega-3, that circadian genes clock and RNTL2 were upregulated, and mitochondrial cytochrome oxidase and other mitochondrial genes in patient number 14 were upregulated. In the next slide, we show the effect of omega-3 on transcription of glycolytic enzymes, and notice that the effects include upregulation of HK1, GAPDH, PGK1, PKM, and LDHA, as well as citric synthase, OGDH, and NAMPT. This is true for 2 SCI patients, 1 MCI patient, but not everything is true for patients with dementia with Lewy body. Omega-3 have heterogeneous effects on mitochondrial genes in MCI patients. The patients with SCI and MCI had increased cytochrome oxidase, MTCO2. They increased TIMM9 and 10. The patient 9 with DLB had downregulation of all mitochondrial genes. Thus, strong effects of omega-3 in MCI contrast sharply with no effects in DLB, supporting specificity for MCI. Now here we show in this complex slide we show single cell sequencing with energy pathways upregulated by omega-3 in the single blood cell transcriptome. Under A, we see clustering of single cells using Sera software. Under B and C, we're showing the shift of gene expression in supplemented patient closer to controls. You can see the results at baseline in patients in red, after treatment with omega-3 in green, and the controls are in black. And under D, we can see the pathways which are improved in supplemented patient, which is the row indicated S. These results show that the monocytes and NK cells have the strongest result with immune system interferon gamma response and glycolysis, hemostasis, cell cycle. Now the TNF alpha inflammatory signaling via NF-kappa B is downregulated by omega-3. In addition to that, in E we can see the effects on individual genes in glycolysis, OXPHOS, and the TCA cycle. And you can again see that in NK cells and monocytes are the strongest result in a TCA cycle, OXPHOS, and nicotinamide synthesis. In this slide, we're showing you the role of glucose as energy for phagocytosis. Phagocytosis is optimal when glucose is at physiological concentration, which is indicated in panel A as 100%, and this equals to 5.5 millimolar, the usual concentration in the blood. When we increase the concentration into hyperglycemic range as high as 300%, or when we decrease glucose to 10%, the phagocytosis is strongly limited, strongly inhibited. This is true in 2 patients, although the results are somewhat different. In panel B, we're showing that the combination drink smartfish resveratrol has the most results with phagocytosis, whereas the single molecule such as DHA, resolvin D1, resveratrol are less efficacious in this particular patient. We tested phagocytosis of amyloid beta in green by macrophages of Alzheimer patients in medium with hypo or hyperglycemic concentrations. Amyloid beta phagocytosis was suppressed in hypoglycemia. Omega-3 addition to the medium with hypoglycemic glucose as low as 10% was able to recover phagocytosis by patients' macrophages. We also showed that phagocytosis was suppressed in hyperglycemia, and omega-3 recover phagocytosis by patients' macrophages in hyperglycemic medium as high as 280%. This is important for diabetic patients who are at increased risk of Alzheimer's disease and infection and do not probably clear amyloid beta at nighttime when their glucose can be either abnormally high or abnormally low. We showed that patients who are supplemented with omega-3 recover phagocytosis in IMDM medium with metabolic inhibitor iodoacetate, which blocks glycolysis. The results of phagocytosis in supplemented patients are shown in black bars. The results in control supplemented with omega-3 in gray bars. In contrast, patients who are, controls which are not supplemented are in bars which are empty and show that there is very inhibited phagocytosis in those macrophages. We tested bioenergetic effects in PBMCs of omega-3, as shown here. We tested oxygen consumption rate, and this was done with PBMC incubated with either negative supplement or with smartfish or with algal DHA. In supplementation with smartfish res, we increased oxygen consumption rate, and we increased ATP-linked oxygen consumption rate significantly more than with algal DHA. We also tested the effect of adding carnitine to omega-3, and we noticed that carnitine increased energy production as OCR further than omega-3 by itself. The extracellular acidification rate was not increased in this study. We compared clinical results in patients with SCI, MCI, and dementia with Lewy bodies. The important result is that patients who were in the study a year or less actually improved their mini-mental state over this period. Patients who were in the study very long time, like patients 12 and 14, improved in the first year, but had a minor change over 5 years. They only lost 0.1 point or 0.05 point, as you can see in patients 12 and 14. On the contrary, the patients with diagnosis of dementia with Lewy bodies had profound decline of mental state. Despite supplementation with omega-3, they lost 1 point per month. This shows the specificity of the effect of omega-3 in Alzheimer's disease compared to dementia with Lewy bodies. Okay. Omega-3 had effects on genes related to lymphatics. In 4 patients, we are showing the effects in 3 patients with diagnosis of SCI or MCI, whereas in the patient with DLB, the effects were not as strong. We're showing the effects of omega-3 on VEGF-C, which is the lymphatic gene. We show the effect on PDPN, which is also the lymphatic gene. We show the effect on ASS1, which is important for macrophage function. We show the effect on AGRN, which is agrin, which is important for the astrocytic end feet. We show the effect on syntrophin, and we show effect on MGAT3, which is a glycosylating enzyme, and then fibroblast growth factor receptor also. So these effects are in the line of supporting the G lymphatic system clearance of soluble amyloid beta. In summary, clinical benefits of omega-3 for patients with mild cognitive impairment include increased energy for glycosylation, for phagocytosis and other functions. They are signaling to increase glycosylation, possibly involving receptors on immune cells, including toll-like receptors and scavenger receptors. They improve cognition in the first year of omega-3 supplementation, and they extend protection against dementia up to 4.5 years beyond cholinesterase inhibitors. These effects are individual and depending upon basal condition. Interesting that omega-3 supplementation increased CD8 T cells, which are the antiviral cells, which have effects against viruses such as coronavirus 2. We acknowledge the collaborators, the participants, and the facilities. All tissues and cells are from the blood samples of patients and brain tissues of Alzheimer patients autopsied at UCLA. Our lab studied immunopathogenesis, macrophages, function, macrophage upregulation of energy genes, blood-brain barrier constructed with human brain endothelial cells provided by University of Arizona. Pellegrini lab performed RNA sequencing and in some cases DNA methylation and epigenetic of AD patients. Young lab showed single-cell sequencing by Jessica Ding. Paul lab helped us with upregulation by omega-3 of circadian rhythm gene RNTL2. Fowl lab performed mass spectroscopy of omega-3 effects on ATP and NAD production. Orion lab showed ATP-linked oxygen consumption rate in macrophages performed by Linda Stiles. And currently we are working with Biswa Choudhury at UCSD, who is working with us on glycosylation of macrophages and peripheral blood mononuclear cells from patients in order to clarify how omega-3 work in patients with hyperglycemia. Thank you.